Welcome to the short video, which is a chance to think a little bit about some tips for better learning, particularly of microbiology at medical school, and a few things which might be useful for other areas of the course as well. And this is my take on explaining what we mean by heuristics. And it's certainly a word and a term that comes up a lot in my teaching, particularly in the early years of the course. A lot of microbiology is taught by heuristics, and there's a lot of value in doing that. What do we mean by that? Heuristics is kind of the term which basically means a rule of thumb. Okay, so it's usually used and applied to mental shortcuts. You learn basic rules and properties of particular areas or groups of microorganisms, for example, and in doing so, you learn a little bit about a lot of microorganisms. And it's particularly well suited for microbiology because of just the sheer vast quantity of organisms which are out there that junior doctors and medical students need to know about. So in my teaching, you'll have encountered this particular image before, the tree of life. And it's a great way of kind of demonstrating the challenge we've got when thinking about microbiology because of the sheer number of organisms which are useful. So you've got enormous areas of life, which some of them are more useful than others, but there's a lot here that can be useful for medical students to know about. And the challenge is, how can you go about it in a sustainable, in a kind of systematic way that's going to be efficient for your learning, but also isn't going to be a kind of just turn into just generating a bunch of lists. And this is a real problem. I mean, the things appearing on the screen are all really medically important, and yet they're all quite distinct from each other. I mean, certainly on the face value. So you've got a mix of bacteria, viruses, and other things, and you can look at them and you can go, well, what does a medical student need to know about these things, and how can I organise my learning to kind of take something from them? Because they're all important, and it doesn't matter whether you're a medical student, a junior doctor or a senior doctor. There's value here. You can also organise them by lists if you want. So here we are, we've just organised them randomly by lists, and we can start off by going, right, Staph aureus, that's really important. So we'll do some Staph aureus infection signs and symptoms. But if you do that for one, then we should do it for all of them, right? About disease profiles, yeah, sure, we could do some disease profiles. We could learn about clinical management one at a time, ditto for treatments, and so on and so on. And the problem with organising it this way and just generating a fairly random list is that it's really boring, it's really inefficient, and it's really unsustainable. So even with the best of intentions, you'll start off really well, but then very quickly the quality of the work and your attention span fades, and then it just doesn't become a particularly useful exercise. And it's not sustainable because what ends up happening is you're faced with enormous lists which you can't possibly maintain for months and years, and forget it. Is there another way? So a kind of a saying that's often heard in medicine is, if you hear the sound of hooves, you think horses, not zebras, meaning basically common things are common. So there is a lot of value at learning about things that are going to be causative agents of particular infections seen in your region, in your particular placement that you're on, whether you're in the UK, whether you're elsewhere in the world. It's a fairly obvious thing to say, but I'm always surprised by the number of medical students that, certainly in the UK, focus on the weird and the wacky rather than the things which, dare I say it, might be more common, a bit more boring, dare I say it. But actually, there's an awful lot of value in going ahead and learning about those particular things. So learning about common things on particular placements when you're learning about a particular case, there's a lot of value. The same is true for context-dependent infections. So if you're on a respiratory placement, learning about common respiratory tract infections is always going to be useful. Is there another way? Well, we can demonstrate this in a kind of another analogy by thinking about life on Earth. So here I've just picked a bunch of random animals and insects and other stuff. And again, we could go about the same thing. So we could look at these things and we could organise by lists, and we can certainly ask some very sensible questions. So we can think about common names. So learning Latin names is useful because whether you're in the UK or the United States or France or Spain or Italy, you can call the same thing by the same name, so universal name. You can think about its relationships with each other, the type of diet, where it's found, geographical spread, all these kind of things are useful. But again, the problem is if you organise them as lists, the same thing is going to be seen. Your attention span starts off really good and you've got great intentions, but very quickly fades and things just drift off. And what you're learning about things at the bottom of your list is no longer useful compared to those at the top. So what we do with heuristics is we start to group things together. We learn some of the main rules and principles around specific groupings that we make. And that means that we know a little bit about all of those creatures, be it macroscopic animals, whatever. We know something about all of them. For example, we could group things by mammals. We could ask questions like, what's a mammal? What's the metabolic differences between a mammal and an insect, for example? Are there cell biology features? In contrast, what features distinguish a non-mammal? And what it means is, even if we were to say discover a new life form tomorrow, and we were to say this is another mammal, we instantly know something about it. It doesn't matter whether it's been studied to death or not. We know immediately, it's a mammal, therefore it's going to have the following features. Let's think of another example. We could think a little bit about grouping by special adaptations, and that in itself picks out some useful learning from. So we could think a little bit about flight. So here we've got a bat and a butterfly. They're not genetically related to each other, but they have common features, i.e. they've evolved the same abilities. And that in itself allows us to kind of think about rules around evolution. And again, certainly within microorganisms, you see similar sorts of behaviors and abilities are sometimes shared between very distantly related things because nature's pushed them to evolve the same sort of abilities. Let's think of another example. So we could think about adaptations to specific habitats. We could think about creatures which are freshwater, saltwater, capable of both. And we can go on and on and on, by the way, but learning about these heuristics means that we can start to learn general rules. The challenge with heuristics is, or rather the kind of thing to always bear in mind, so there are always going to be things that are exceptions to the rules. And that is, I suppose, the main disadvantage. But as long as you're aware of it, it's quite a useful thing to be mindful of. Okay, so let's now think a little bit about how we can apply heuristics to some specific microbiology. Let's go back to our list. This was just the randomly generated list of microorganisms from before. So we can do the simplest thing, and we can group them according to the type of microbe. There are 4, broadly speaking, main medically important groups of microorganisms, so we can group them together. So we've got on the top left, we've got viruses. On the top right, we've got prokaryotes. We've also got eukaryotes. We can split the eukaryotes up a little bit further, so we can think about those which we might consider to be parasites, so quote unquote parasites. So they might be the kind of the worms, the protozoa, so things like plasmodium, but also the fungi as well. And the fungi can further be split down into molds and yeasts. So that is in itself a useful way to start. And by comparing and contrasting all these different things, and a lot of it is going to be very basic revision from biological concepts you've addressed at school, that's going to be a really useful place to start. So viruses can basically be grouped according to a number of different ways, but there are only really a couple that are useful for medicine. We can group them according to whether they've got an envelope or not. So SARS-CoV-2 or norovirus are good examples of enveloped and non-enveloped viruses, and they have different infection control, or there are some infection control differences between those two groups. We can think a little bit about their hereditary material, so RNA, DNA, and then also we can then go on even further to talk about whether, for example, the RNA is segmented or not. We could talk about its sense, so whether it's a positive or a negative sense strand. We could think a little bit about whether it's a single-stranded or double-stranded molecule. I suppose we can go on, but these are just some examples by which you can group and generate heuristics around learning about these things. Prokaryotes. So we can divide the prokaryotes into 2 groups. We can think a little bit about the bacteria. We can again group these according to their cell wall, the way they respire, what I've called their lifestyle. So cell wall structure can again further be broken down to those 3 things. Atypicals, we can break that even down even further. And then we can start considering respiration. So the idea that there are some that can grow with and without oxygen, and facultative anaerobes that sit in the middle. They can actually cycle between potentially growing with oxygen and without, as and when the need arises. Archaea are an unusual one because they are environmentally an important group, but medically not that much of a priority, to be honest. So that you do find them in the body, but they're not all that useful. So they're not, I would suggest, anything you should be spending a huge amount of time learning about. And then we come on to the eukaryotes. So we've got, again, the fungi and the parasites, and these are kind of ideas that we start to introduce more in year 2, and certainly in the UK, these are probably less common than your viral and bacterial infections that you'll encounter a lot more frequently. Though arguably, certainly fungi are becoming more and more of an issue, and parasites should always be mindful, or you should always be mindful, dependent on, for example, travel history from someone you're examining. The last thing to consider is whether this sort of approach, these kind of focusing on these broad ideas, can be applicable to other areas of the course, particularly those quite early on in the course. And I'm going to argue that yes, they actually probably can. And there are a couple of quite useful things to say with respect to case-based or inquiry-based learning. Let's imagine that you're looking at a case of pneumonia in an older gentleman. Okay, so let's imagine we now go ahead in our group and we set a bunch of very specific questions which focus right in. So we might consider some of these to be quite smart questions. So there might be specific things we can go away and look at, do lots of research on. So we could focus all of our questions in on specific causes of that pneumonia. We could think about specific complications, particularly if there are any that are related to that case. We could think about generating a top 5 list of causative names of things which might be really specific, particular antibiotics that maybe this person may have been prescribed. And again, we could look at mode of action of those particular drugs. The problem is that if the questions are almost too specific, it's too specific to that particular case. And also, it doesn't really generate much in the way of learning or discussion within the group. And so sometimes what's helpful is to zoom out. And rather than kind of just do all of your questions focused, step back and start to think a little bit in the back of your mind about heuristics and those bigger picture ideas, thinking about anatomy, thinking about what are bacteria and viruses and how does the immune system differ in its response between the two things. And these are going to feel very basic, but when we talk about going back to basics, this is really what we're talking about. It's about going back and revisiting and reapplying and learning. Because imagine that the next case was pneumonia in a young child. How would your very specific questions apply in this new setting? Well, probably most of them would not actually be that useful. They'd be great if another 75-year-old man walked in with pneumonia, but they wouldn't be useful at helping if it was in maybe the same condition, but in a different demographic. So now you're going to have to generate, if you want to go and make more specific questions, the same sorts of things, but just applied to a completely different demographic. Whereas your broader questions that you'd asked before also apply here. So previously, with the analogy of what's a mammal, and you discover a new mammal the following day, and you know immediately something about it, here, because you've revised and you're quite familiar with respiratory tract anatomy and differences between immune responses, and you've got basic principles around antimicrobial therapy and all the rest of it, you already know when someone walks in through the door something about the condition. Because your research is focused on broad ideas that you've discussed, it means your questions are not too specific. And by the way, just discussing those things and explaining things together is a really great thing to do together as a group. And we can take this on a step further. We can think about what if the next person who came into your clinic had COVID-19 or had asthma or had COPD or some other respiratory condition. Well, again, these top kind of higher order ideas don't always apply, but actually they're not bad places to start in helping understand some of the body's response and trying to understand the pathophysiology of what's going on. Anyway, just a few ideas to get you thinking. Naturally, any questions, get in contact.